Science, Grade 7, Unit 10. The correct use of the microscope. A bit from the history. The compound light microscope was first made by Zacharias Janssen. The magnification power of it was 9 times and it was similar to a toy telescope. But the honor of making a microscope first goes to the Dutch scientist Antony van Leeuwenhoek. The reason is that a world not seen from a naked eye was revealed by this instrument. Anton van Leeuwenhoek, father of microbiology, he made his own lenses, some as powerful as the microscopes we use in class. First person to see living microorganisms. In this picture, I have shown you a picture of the microscope made by Anton van Leeuwenhoek. What are the objectives of this lesson? By the end of this unit, you will be competent to observe small things using transparent objects with convex nature. Use light microscope. Identify and name the parts of a light microscope. Calculate the magnification power of a microscope. Observe some specimens prepared by you. These are the subunits. 10.1 The simple microscope. 10.2 Magnification of a microscope and resolving power. 10.3 Compound light microscope. 10.4 The electron microscope. Observe minute objects. Microscopes are used to observe objects that cannot be seen with our naked eye. Telescopes are used to observe distant objects. 10.1 The simple microscope. Magnifying lens, hand lens. These names can also be used. Now I have shown you a picture of a simple microscope or hand lens. You can see a convex lens. I have showed you a convex lens here. This convex lens is fixed to this frame. Now this is the frame and this is the convex lens and the handle. Now this is the label diagram of a hand lens. Now you can see the convex lens here again and the frame and the handle. I think you all have used this type of hand lenses to observe parts of flowers, parts of animals and some other instances. Simple microscopes are used to read letters on a currency note. You know what currency notes are? And to read pharmaceutical labels because the letters on pharmaceutical labels are very small. We cannot read. So we can use a simple microscope to read those labels. And we use hand lenses to observe parts of flowers, insects and soil particles and also to observe fingerprint. So you can add some more instances where we use simple microscopes, the instances where we use hand lenses. The distance between the object and the lens is an important parameter to get a clear image with maximum magnification. There should be a constant distance kept between the object and the lens when an object is observed by a lens. 10.2 Magnification of a microscope and resolving power. What is magnification? An object when enlarged several times of its actual size, it is referred to as magnification. Or we can take it like this: the number of times a specimen is magnified is known as the magnification. The number of times a specimen is magnified is known as the magnification. What is resolution? I think before starting this lesson, if you have the textbook with you, it's better. Right, if you have the textbook with you, you can turn to page number 145, activity 10.5. For that activity, you need a white paper, a pencil or a pen, two hand lenses. You can have hand lenses, you can buy from some bookshops, and you have to have two hand lenses. It's better to have hand lenses with you if you want to see, observe the environment. Right, what is the method? Mark two dots with a distance of about 0.5 millimeters. Mark two dots with a distance of about 0.5 millimeters. So you can use the ruler to mark the two dots. Observe the distance between the two dots and their magnification by using a hand lens. One hand lens. Now use two hand lens to observe the distance between the two dots and their magnification. Right? There is another activity given. For that activity, you need a colorful picture from a paper, a hand lens. Observe a picture from the paper by a hand lens. You have to present your observation to the class, or when you go to school, you have to present your observations to the class. That is the activity 10.4 given in your textbook. You will observe that the respective image is composed of a large number of small dots seen by the naked eye. You will see these dots very clearly by a hand lens. Now you will realize that the resolving power of the hand lens is higher than the resolving power of the naked eye. Normally, there should be at least a minimum distance of 0.1 millimeter between two adjacent points to distinguish between them by the naked eye. Now you will realize that the resolution of the naked eye is around 0.1 millimeters. Resolution is the minimum distance by which two points must be separated in order to be seen as two distinct points. Maximum visibility for a human being with the naked eye is about 0.1 millimeters. The resolving power of the hand lens is higher. That's why we use hand lenses to observe small things. The resolving power of the hand lens is higher than the resolving power of the naked eye. To understand that, as I explained earlier, you have to do activity 10.4 and 10.5 in your textbook. Resolution. It is possible to obtain a higher magnification and higher resolution by combining two convex lenses or hand lenses. And there is a problem. The image is inverted upside down when two lenses are combined. There is no issue if we combine two lenses. We observe animal parts and cells, plant cells, animal cells, and also no issue if it is upside down or inverted. We can observe them easily. Compound. Now we are going to discuss about compound light microscope. There are two more names given to this one: light microscope, optical microscope. We can use light microscope, optical microscope. These names can also be used. This microscope is used to observe fine things which are not visible to the naked eye. Where do we use these type of compound light microscopes? Normally we use them in our school laboratories, in hospitals, in universities and medical research institutions, medical centers, pathology laboratories and plant research institutions. And it is used to observe plant cells, animal cells and microscopic animals like amoeba, paramecium, euglena, right? And plants very small plants, and also to observe crystalline objects like sand particles, clay particles, like that. Now, what are the parts of a microscope? In your textbook, they have given a picture of the compound light microscope. Now, go through this. You can see the eyepiece is here, the body tube. This is called the body tube, and this part is called the nose piece. The objective lenses, there are three objective lenses. They are fixed to the nose piece, and then you can see the coarse adjustment, and this is called the limb or arm, and this is the coarse adjustment, this is the fine adjustment, right? And this is the foot, and you can see the stage here, and a slide is kept on the stage, and clips are used. Here are two clips which are used to fix the slide, and below the stage you can see the diaphragm and filter and condenser, and a concave mirror or a plane mirror is fixed here. It's a light reflector. We use light energy as the source. So we use a concave mirror or a plane mirror to get light. These are the main parts of a compound light microscope. Now, the lens kept closer to eye is called eyepiece. The lens kept closer to the specimen is called objective lens. I will show them again. Eyepiece. Now, this is the eyepiece, the closest lens to the eye. It is a convex lens. Here, the convex lens is fixed. Normally, the magnification is shown as 5 times, 10 times or 15 times. The specimen is magnified by using them as 5 times, 10 times or 15 times by using eyepiece. The objective lens, there are 3 lenses fixed to the nose piece. The closest lens to the specimen. Now, if we keep a plant cell or animal cells here, they are the specimens, or parts of insects, they are the specimen. The closest lens to the specimen. Now, these are the closest lens to the specimen. They are made of convex lenses and it magnifies the specimen. Mostly there are 3 objective lenses as I mentioned earlier. There are 3. The first one is lower power objective lens. The magnification is 4 times, 5 times or sometimes 8 times. Mid power objective lens, the lens with the medium magnification, normally it is 10 times. And then comes high power objective lens, the lens with the highest magnification, normally it's 40 times. And you can see the coarse adjustment here to adjust the body tube. If you move this, rotate this, you can move the body tube to get a clear image of the specimen. And there is another adjustment called fine adjustment to adjust the body tube finally to get a clearer image of the specimen. And stage to keep the slide of the specimen, and clips to keep the slide of the specimen on the stage. And there is a part called diaphragm. It controls the amount of light obtained by the specimen. And there is a mirror, maybe a concave or a plane mirror, focuses light onto the diaphragm. It focuses light onto the diaphragm. And there is a part called base. It is used to keep the microscope on the supporting surface steadily. The lens kept closer to eye is called eyepiece. The lens kept closer to the specimen is called objective lens. It is possible to get a magnification of about 2000 times by using compound microscopes. The magnification of a compound microscope is obtained as a product of the magnification of the eyepiece and the objective. We can use this equation to get the magnification. Magnifying power of the objective lens into magnifying power of the eyepiece. Shall we use that? Now look at this. How do we find the overall magnification of a light microscope? Here, this is the eyepiece and these are the objective lenses. Now, if eyepiece magnification is given as 10 times, objective magnification is given as 4. So, 10 times 4 is 40. The overall magnification is 40. If the eyepiece is 10, objective magnification is 10. 10 times 10, 100. Eyepiece magnification, if it is 10, objective magnification 40, the overall magnification is 400 times. Eyepiece magnification is 10, objective magnification is 100, so overall magnification is 1000. So that's how you have to calculate the magnification of a microscope. Components of a microscope could be classified under 3 systems according to their usage. What are they? Photo system, mechanical system, body system. Photo system, mechanical system, body system. Photo system consists of the eyepiece, objective lens, mirror, a source of light. Sometimes electric bulbs are fixed to those microscopes. You may have seen. Mechanical system consists of adjustable body tube, stage adjustment, you can move the stage, diaphragm adjustment and stage clips. These stage clips belong to mechanical system. Body system consists of the handle or arm, stage, body tube, pedestal or base or foot. Pedestal, base or foot. The correct method to use a compound light microscope is given in your textbook page number 148. The correct method to use a compound light microscope. First, keep the microscope on a vertical table steadily in a place where strong sunlight will not fall. You have to get the instructions from your teachers as well. Use the rough adjustment or coarse adjustment to bring the lower power objective lens down. Adjust the mirror and the diaphragm to obtain a clear spot of light by keeping the eye open on the eyepiece. When you observe using the eyepiece, you have to keep one eye and the other eye must keep open. Fix the prepared slide on the stage using clips. Bring the lower power objective lens upward using the rough adjustment until a clear image is obtained, making sure that both eyes are open during the process. Keep the comfortable eye at a distance of 1 cm to the eyepiece. Try to obtain a clear image using fine adjustment. A sharp and clear image could be obtained by adjusting the middle and high power objective lens if necessary. Use the rough adjustment to bring the lower power objective lens upward after the observation. Remove the specimen from the stage. Use a dry piece of cotton cloth to wipe the stage. Now, if you prepare a slide using a piece of onion, you will be able to see the cells like this. You can see the plant cells under the compound microscope like this. You will be able to identify the nucleus the cell wall. Now this is the cell wall, and the cell membrane. Inner to the cell wall there is a membrane called cell membrane, will be able to observe, and there's a large vacuole in plant cells. So you will be able to identify these parts in plant cells. Shall we go through it again? You will be able to observe nucleus in plant cells under the compound microscope. You will be able to identify cell wall. Inner to the cell wall there is a membrane called cell membrane, and the vacuole will be able to identify. Animal cells under the compound microscope. Now these are cheek cells taken from a mouth. Using a yogurt cup, you have to take some cells from your mouth, cheek cells. So you will be able to observe the cheek cells under the compound microscope like this. Cell membrane, you will be able to identify the cell membrane, the nucleus and the cytoplasm, the jelly-like substance in the cells. Cell membrane, nucleus and the cytoplasm will be able to identify in animal cells. Now I have shown you a slide of epidermal cells taken from a leaf. This is a dicot leaf showing open stomata. Right, now you can see the epidermal cells like this. The slide will be like this. If you draw the diagram, it will be like this. Normal epidermal cells can be seen, and you will be able to identify guard cells like this. Now these are the guard cells. In between the guard cells there is a pore called stomata. Stomata. You will be able to identify stoma, guard cells and epidermal cells. Now this picture, this slide shows plant cells under a compound microscope. Now rohioepidermal cells will be able to observe like this under a microscope. Nucleus, secondary cell walls and the cytoplasm and some pits. When you go to the higher grades, you will be able to study more about these parts. We are going to discuss about the electron microscope. To view small objects even more closely, scientists use electron microscopes. These microscopes use beams of electrons instead of light to magnify objects, not like the compound microscope. There they use light. Here we use a beam of electrons. Electrons are some of the particles or bits that make up atoms. Atoms, you can see the electrons in atoms. Electron beams cannot travel far in air. Objects must be put in a vacuum. Now, we have to keep the specimen in a vacuum if you want to observe using an electron microscope. Vacuum is a place. It's an airless space before they can be seen with an electron microscope. Electron microscopes can magnify objects up to 1 million times. This magnifying power makes electron microscopes very important scientific tools. However, they cannot be used to study living things. This is a very important point. We cannot observe living things under an electron microscope because living things cannot survive in a vacuum. Living things cannot survive in a vacuum. That is a very important point. Now I have shown you a picture of an electron microscope. See how huge it is, right? In your textbook also they have shown you a picture of an electron microscope. In your book, the instances where we use electron microscope to observe the activity of pathogenic microorganisms like viruses, bacteria during research. to learn about internal structure of the cell in detail used in genetic research. They have given some instances where we use electron microscopes. What are the advantages of electron microscopes? They give very high magnification. The maximum magnification power is 500,000 times. Incredibly high resolution. The maximum resolution is about 0.0005. Material rarely distorted by preparation. It is possible to investigate a greater depth of field and diverse applications. These are the advantages of electron microscopes. Electron microscopes are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, biopsy samples, metals, and crystals. Industrially, electron microscopes are often used for quality control and failure analysis. Science of microbiology owes its development to the electron microscope. Study of microorganisms like bacteria, virus, and other pathogens have made the treatment of diseases very effective. Now here I have shown you a picture of coronavirus, and different shapes of viruses are shown here. Different shapes of viruses are shown here in this picture. The transmission electron microscope. Now these are the parts of the transmission electron microscope. You can see, you can go through this slide and try to understand the parts of the transmission electron microscope. And next, scanning electron microscope. This is the structure of an electron. Scanning electron microscope. The specimen is kept here. Stage is shown. Secondary electron detector is here. And electron gun is shown here. Electron beam, anode, magnetic lens are shown here. Backscattered electron detector is shown here. Now, the worksheet, the correct use of the microscope, unit 10. You have to name the parts of the compound light microscope given in the diagram. What are the optical equipment which can be used as J? State the use of A. State two things which should be considered when handling the compound light microscope. Part B1. Following are the steps of handling a compound light microscope. You have to read and write the English letters in the correct order. Place the prepared glass slide on the stage. B. Using the coarse adjustment, lower the low power object to get a clear light patch. C. Place it stationary on a horizontal table. D. Obtain a clear image using the fine adjustment. E. Observe with a convenient eye through eyepiece while both eyes are open. 2. Briefly describe what is magnification. 3. If magnifying power of eyepiece is 5 times and the magnifying power of objective is 40 times, find the magnifying power of the microscope. Electron microscope is an advanced equipment than compound light microscope. State 2 advantages of electron microscope. State a disadvantage of electron microscope. Select the answers from the words given and fill in the blanks. Eyepiece, resolution, convex base, electron microscope, cannot be, transparent, desiccator, diaphragm, magnification. The lens kept close to the eye in a microscope. Blank. A blank lens with a handle fixed onto it is called a simple microscope. 3 is the maximum distance by which 2 points must be separated in order to be seen as two distinct points. Number 4, the blank helps to keep the microscope on the supporting surface steadily. 5. Microorganisms blank observed from the hand lens. Number 6. Blank objects having a convex nature can be used to magnify small things. Number 7. If the microscope is not used for a long period of time, the lenses should be removed and placed inside a blank. Number 8. To observe viruses, blank can be used. 9. Blank controls the amount of light obtained by the specimen. 10. The number of times a specimen is magnified is known as the blank of the lens. And in your textbook, they have given factors to be considered in the use of microscope. Number 1. When the microscope is carried from one place to another, the arm of the microscope should be held by your familiar hand and the other hand should be kept below the base of the microscope. Then hold the microscope towards your body. Number 2, wipe the stage using a clean piece of cotton after using the microscope. You have to protect the microscopes. Number 3, if the microscope is not used for a long period of time, the lenses should be removed and placed inside a desiccator with silica gel which absorbs moisture or anhydrous calcium chloride. Silica gel and anhydrous calcium chloride, both these chemicals absorb moisture. So we have to keep the lenses inside a desiccator when the school vacation comes. The microscope should be stored in an upward position to minimize the accumulation of dust. Number 5, use clear slide and cover slips in the observation. Number 6, do not change the lenses of one microscope to another. That is very important. Do not change the lenses of one microscope to another. So, before doing the worksheet, it's better to read the textbook and start to answer the questions. Thank you and please do subscribe to my channel.